My project is on the Alpha Epithelial Sodium Channel, or Alpha ENAC. It is a membrane-bound ion channel that moves sodium ions. This aids in reabsorption into the bloodstream. It is found in epithelial cells, or cells on the surface of body components like organs. These cells are found in the distal convoluted tubule within the nephron of kidneys, which are a part of the urinary system. Functions of the urinary system include eliminating waste, regulating blood volume and blood pressure within animal organisms. This is my model of the epithelial sodium channel. These green balls represent sodium ions. The blue structure represents a phospholipid. The purple, pink, and orange balls represent ion channel gates, and the gamma, beta, and alpha structures represent the different ENAC channels. When the channel is in a closed state, the sodium ions are not able to pass through the ENAC channels. Channels are pathways that can be open or closed to allow for the intake of materials into a cell based on the conformation or shape of the molecule. When the channel is open, the sodium ions are able to pass through. This occurs in both the mutated and the normal ENAC. Both the mutated and normal alpha epithelial sodium channels have open and closed mechanisms. The difference occurs in the gate of the mutated alpha epithelial sodium channel. The normal protein has the amino acid cysteine, which has a sulfhydryl functional group. Two sulfhydryl groups can interact and form a disulfide bridge. This helps stabilize the protein structure. Its side chain is polar and hydrophilic. The mutated protein switches cysteine with arginine. It is basic and has a positive charge. Unlike the original protein, it forms ionic bonds rather than disulfide bridges. Because of the structural difference caused by a lack of disulfide bridges, the gate has an increased open probability, meaning it is open more often than the normal channel. This allows for sodium ions to pass through more often, thus increasing the amount of sodium reabsorbed into the bloodstream. It is important to keep in mind that the epithelial sodium channel has a beta, gamma, and alpha region. This mutation occurs in the alpha region as depicted in the model. This protein has an increased amount of activity when the mutation occurs where cysteine is replaced with arginine. The amount of sodium reabsorbed into the body increases. The fluid volume of sodium is involved in the regulation of blood pressure in the organism. Water in blood has a high retention in blood in response to an increased presence of sodium ions. This results in hypertension. Liddle Syndrome is the result of salt-sensitive hypertension. Hypertension is also a risk factor of stroke, myocardial infarction, heart failure, and ESRD. This model has a few limitations. First, it does not show how the lack of a disulfide bridge leads to a change in the open and closed mechanisms of the channel. Secondly, it does not show the structural difference between the amino acid replaced that was depicted on a piece of paper. Finally, it does not show where in the cell the structure is. It does not show what is on either side of the membrane.